and we'll start right at noontime. So we'll give it another minute or so. So I have noontime Eastern Standard, and to respect everybody's time, we will start on time. Thank you for those of you that are joining us on Facebook Live, through the website, and for those of you that signed up and are taking the time to join us today. I am Krista Rubin, and I want to welcome everybody to AMAT Skin Cancer's webinar series, Skin Cancer in You. I am a nurse practitioner in Boston, specializing in skin cancers, and I am also AMAT Skin Cancer's Ask a Nurse. I am thrilled to be here today to bring you a wealth of information on various aspects of skin cancer treatment, management, and emotional well-being. We've had a great webinar series so far, and today I'm so pleased to have joining me Dr. Kenneth Tsai, who is an MD, PhD, and board-certified dermatologist and dermatopathologist. He's the Vice Chair Research at Moffitt Cancer Center and Research Institute in warm and sunny Florida. Dr. Tsai's research focuses on understanding the genomic changes that drive the development of non-melanoma skin cancers, such as basal cell and squamous cell, and also how they evade the immune system. So very interesting topic for discussion today. So welcome, Dr. Tsai. Thrilled to have you.
Thank you.
We
Thrilled
go
to be
way,
here.
yeah, we go way back. We knew each other, I hate to say it, but a couple of decades ago. So your introduction is actually very appealing to me because the topic of today is the genetics of skin cancer, but really I probably should have titled it The Genetics and the Genomics of Skin Cancer, because genomics is a hot word right now and certainly a hot topic of interest. So I think we should start this by, I'm going to ask you to explain the difference between genetics and genomics for our audience.
Sure, yeah. I think, genetics really, I think the way to think about that is um, first and foremost, what we inherit from our parents, right? So there is a um, sizable, I would say, contribution of what you inherit from your parents, what we call genetics, in determining your obviously things about the way you look, your pigmentation, hair color, eye color, things like that, but also including in skin cancer, your risk for developing cancers such as melanoma, basal cell carcinoma, squamous cell carcinoma, Merkel cell carcinoma as sort of, some of the big skin cancers that we think about. And so there's that component, and then there's also, we can talk about this a little bit as well. Some rare familial predisposition syndromes, and these are of course very very important in cancer. It's actually how many of the major genes that we now know are important in determining cancer risk, but also mechanistically how cancers arise, were really originally identified in families that had very unusual high predisposition to developing cancers at an early age. So we now know that some of these cancer predisposition syndromes, um, uh, for example, can confer exceptionally high risks developing melanoma or basal cell carcinoma, as an example. And so those are really instances where genetics and inheritance come into play in your family history. Genomics is really, in some ways, a kind of a much broader term. It's really about the entire complement of DNA that you have, not only uh, um, in all the cells in your body, but in particular, we focus in cancer a lot on the changes that occur in the cancer cells themselves. And so this can really be, a very wide array of changes that occur in your DNA, not just the string of letters that occur in your DNA, the sequence of letters that occur in your DNA, but also even large-scale chromosomal changes. Um, we um, we have 46, of course, 23 pairs of chromosomes. Um, sometimes these large chromosomes can swap large areas of DNA and actually make new pieces of DNA as a result. And these can also be really important in the development of cancer. So the way to think about genomics is sort of much larger scale changes that occur in the DNA across the entire complement of chromosomes that we have, and how that can be changed in the cancer cells.
Okay, and this is not always an easy topic for everybody because DNA genetics is very confusing. So I encourage anybody in the audience, if you have questions, please submit through the Q&A at the bottom of your screen, where we'll do our best to answer those as they come through. But Dr. Tsai, a couple things that you mentioned that I just want to highlight. One, you used the word familial. And so with regard to skin cancers, that's certainly a question we always ask patients when we're seeing them initially is, do you have a family history of skin cancer? Now, we know that there's a definite connection for patients that have melanoma with a first-degree relative. But could you just comment on the risk of skin cancer, basal cell or squamous cell familially? So in other words, if you have family members with a squamous cell or a basal cell, and I'll remind people that basal cell is the most common skin cancer really in the world, but does that increase somebody's risk because you have a family member with a basal cell or a squamous cell? Could you just touch upon that?
Sure. I would say that the data on that is a little bit less, I would say a little bit less strong in some ways as it is for melanoma. But in basal cell and squamous cell carcinoma, some estimates range around sort of a 2 to 4-fold increased risk if you have a first degree relative that also has that diagnosis. The thing about basal cell and squamous cell carcinomas is that overwhelmingly the risk of developing those cancers is related to exposure to sunlight, particularly ultraviolet radiation, which also incidentally can come in the form of tanning beds and things like that. But over that risk overwhelms all of the other risks that are really associated with the development of these carcinomas, which arise from the outermost layers of the skin. Those are the epidermis. That's the layer of skin that forms the barrier between you and the environment. And so, so overwhelmingly, that's really related to sun exposure. As I was alluding to a little bit earlier, there's some, basal cell carcinoma, for example, has a syndrome that goes along with it. It's called Gorlin syndrome. It's relatively rare. It's estimated to occur in about 1 to 30 to 1 to 50,000 people. And it has a whole host of other abnormalities that come along with it. And they're often, we're very, very interested in whether or not there's an affected parent or first degree relative because obviously it's inherited. You have to have two um, uh, um, uh, different copies of a particular gene to get that syndrome. And that's characterized with very, very early onset of multiple hundreds of basal cell carcinomas that can arise individuals. So it's a very dramatic and unusual phenotype in terms of the patients that getting that number of basal cell carcinomas. Obviously, getting one skin cancer increases the risk of you developing a subsequent one, but Gorlins is very unusual in the sense that patients can get dozens to hundreds, even before the time that they're in their 30s or 40s, which is not typical for most basal cell cancers.
Yeah, and to just highlight a couple of things that you mentioned. So, one being is it's often the behavior of families. So that's why we see skin cancers commonly in families because you may vacation together or, brothers and sisters may play together outside. So we often see that it's more an environmental risk rather than a genetic risk. But certainly for those genetic risks, on top of environmental risks, then you're looking at what you alluded to earlier is that increase in fold of risk of developing a skin cancer. So there's a lot that goes into it. But this whole arena of genomics and just really understanding the inside of the cancer cell and kind of what makes it grow, what makes it thrive is really kind of the area of interest moving forward. Can you comment a little bit about the impact of the evolution of genomics and how it's going to, or how it is affecting cancer care, particularly with regard to skin cancers?
Yeah, absolutely. I think in many ways, I'll start with melanoma, but sort of get into some of the other cancers. So, you know, with uh, melanoma, for example, one of the uh, uh, genes that was first identified as an important in the development of melanoma is BRAF. This is a gene that, when mutated, causes cancer cells to divide. So there's a signaling pathway that gets activated. The cancer cells don't know how to stop dividing, and that's something that contributes, of course, to the development of melanoma and other cancers. So it turns out that this is very important, and we use testing in melanoma, genetic testing in melanoma of the cancer cells themselves to see whether or not this gene is mutated, because um, there are specific therapies that have now been developed specifically to suppress that protein that's made from the defective gene. Um, Another great example is the identification of actually the gene that causes Gorlin syndrome. So that's due to a defective copy of the patched 1 gene. And so that led to a whole um, understanding of the pathways that contribute to the development of basal cell carcinomas. And so that too actually led to the development of a whole class of compounds that specifically inhibit the signaling pathways that are important for creating basal cell cancers. And so understanding these genetic defects that occur in the cancer cells um, actually has been very, very important for the development of new therapies, and very, very effective therapies, I should say, for, as an example, melanoma as well as basal cell carcinomas. One other thing I'll mention, which is a little bit more kind of genomics, which is that one of the things that has been uh, extremely promising, really game changing in cancer care, has been the development of immunotherapy. And the idea there is that um, the immune system has the capacity to recognize that your cancer cells are abnormal. And actually, we now have very, very powerful drugs that stimulate the immune system, sort of release the brakes on the immune system to try to get them interested in killing the cancer cells. And this has worked very, very well for melanoma, squamous cell carcinoma, some instances of basal cell carcinoma. And it turns out that what it's related to is that the immune system can recognize abnormal proteins. And abnormal proteins come about as a consequence of having altered DNA, the DNA mutations that we were talking about, the genetic changes that occur in cancer. And so it turns out that if you have more of these mutations, you're very likely to make more different kinds of mutant proteins or different altered proteins that the immune system has a higher chance of recognizing on your cancer cells. So it turns out that immune therapies actually work better as a general rule in cancers that have very high mutational burdens. In other words, large amounts of of abnormal proteins made by cancer cells. And so that's actually very important for and relevant for skin cancers because, as a rule, most skin cancers, having been driven by or caused by excessive sunlight exposure, often have a lot of DNA changes, and therefore they make a lot of altered proteins, and therefore they are more likely to be sensitive to these immunotherapies. So having a sense for specific changes in specific genes like BRAF and PATH1, as well as having a global idea of how the DNA of the cancer cells has been changed, in particular by UV light, has really, really important implications for cancer care.
That was a great explanation. It really kind of brings it all around. We started with speaking of genetics and brought it around to genomics. And I think that's important for any audience member or family member on this webinar, understanding why immune therapies have really kind of taken over patients with advanced skin cancer, sometimes before surgery, after surgery, and why they work. So it's a case where the more unusual, the better. So the more unusual the proteins are, the immune system is more likely to recognize these. And that's why some cancers don't respond to immune therapies, because they don't have enough mutations for the immune system to identify. But in the case of skin cancer, they do. And I just want to make the point, to highlight this point, that cancer is really just a disease of the DNA. And in this case, for patients with or individuals with skin cancer, the DNA is altered from ultraviolet exposure, from sunlight or tanning beds. So when we talk about protecting your skin, even from future skin cancers, when we encourage clothing and SPF, it's because we want to try to minimize that DNA damage moving forward, which can continually happen. I often get the question, I've already had skin cancer, why bother wearing sunscreen? And I think this brings it around to a great example of why we want to minimize those genetic or DNA changes. So, all right, so can we talk a little bit about who should be getting genetic testing? And maybe actually, no, let me take that question back. Let me ask again, how is a tumor assessed for all of those changes? How do you know if your tumor has all of these genetic changes that you're mentioning? How do the researchers know that? What's the process?
Yeah, so a typical way this is done is that very often cancer is diagnosed from a skin biopsy, right? So we take, a dermatologist might see an abnormal looking mole or an unusual growth on the skin. He or she might be suspecting a skin cancer. It gets sent off to the pathologist for interpretation. And so a dermatopathologist will look at that specimen under the microscope and make a diagnosis of whether or not this is cancer, and if so, what kind is it? And you know, in the scenarios that I was just mentioning, that if, in particular, if these, many early stage cancers can be removed surgically. It's an office procedure, relatively easy to treat. But I think, but in circumstances where we're worried about advanced disease, where it may be an unusually large lesion, difficult to manage surgically, or there's a risk of metastasis, then genetic testing becomes very, very relevant, because now we have to entertain more aggressive therapies, like the therapies I was talking about earlier. These are in many cases either oral medications or medications that need to be infused. And there we will have to know something about what the options are for therapy. And in that sense, we'll want to know about some of the specific genetic mutations that are that are present. So in that circumstance, we can now take the specimen that has been biopsied. We can actually extract the DNA from that tumor biopsy and sequence it. So we can actually basically assemble the entire DNA a complement of that tumor and really understand, is there a BRAF mutation if it's a melanoma? Is there a patch one or some other hedgehog pathway mutation in basal cell carcinoma that would suggest that that cancer is sensitive, would be sensitive to, one of the therapies that we have available. And actually, sometimes, um, tumors can be very, very unusual, right? Because as we've just noted, um, they can be formed, from a lot of DNA damage that has occurred and mutations, and sometimes what kind of cancer it is actually can be very difficult to tell. And so sometimes genomic testing is used on the cancer specimens to help us even diagnose the cancer, to make sure that, hey, this really is a melanoma, or this is really a basal cell carcinoma or a squamous cell carcinoma. And one of the ways that is interesting in terms of how we do that is that because many skin cancers are related to UV exposure, there's a specific type of mutation that UV causes in the letter sequence of DNA. And so that is actually detectable through sequencing, and we can actually determine with a reasonably high degree of certainty nowadays whether a cancer has been caused by UV exposure or not. And many times that can help us with actually even diagnosing and identifying that this cancer is actually truly coming from the skin. So that also has implications for how we take care of it in terms of what therapies we use.
And so I think it's important to make the point that there's different types of testing. There is genetic testing when you're trying to determine if your particular type of cancer is a genetic or familial form. In other words, women that may have breast cancer have a gene that's mutated known as BRCA. And that is important to identify for family members because that is something that's familial. But the testing that you're alluding to now within the tumor itself, that's a different type of testing. Could you just comment on that? Because I know that that's confusing for patients that have melanoma when I see them, and we have a discussion about the BRAF mutation. It's still a difficult concept to understand that it's not the person's BRAF mutation, but it's the actual tumor. Could you just clarify?
Sure, you bet. So I think, a lot of the molecular testing we do now is on the cancer cells themselves. So on the skin biopsy that you've had, we're really interested in identifying what are the specific DNA changes that have occurred in the cancer cells. I would say that maybe a little less commonly, if we're suspecting that, you may have inherited a genetic, a predisposition, that you may have inherited something from your parents, um, you know, um, because, for example, you've gotten, many melanomas, you have, uh, a lot of dysplastic nevi, or you're one of these individuals that has dozens and dozens of basal cell carcinomas, and you have some of the other characteristic findings where there's a suspicion for that. Um, we may test normal cells actually from you, and that can be sometimes skin fibroblasts or blood, where we want to understand, are there changes in your DNA that affect all the cells in your body and may have contributed to a risk of developing cancer? So I think that's what Krista's referring to, which is that um, we're really asking two different questions. In one setting, we're really trying to figure out, okay, what are the specific changes that might be um, in the cancer cells themselves, and then in another setting, the genetic testing setting, trying to understand um, what changes in your DNA might have been inherited, from your parents that might actually predispose you to developing a cancer. So different tests asking somewhat different questions um, about uh, sort of different aspects of the cancer journey, if you will, but really answering different questions about the cancers themselves, but also having very important implications for how how we not only treat the specific cancer, but how we keep track of patients. Um,
Yeah, we actually have some great questions coming in, and I
yeah.
think we probably answered one of them. But one of our members asks, who should get tested for genetics for developing skin cancer? Kind of similar questions from another member, audience member here. But if you're at high risk for skin cancer, can you get genetic tested?
Yeah, I mean, I think I think it's important to remember that the overwhelmingly, for the vast majority of people, the biggest risk factor for developing most skin cancers, in particular the basal cells, the squamous cells, which are much more common overall, is really about exposure to UV and being, it's about, sun safety and that exposure. I think that where it becomes important to think about genetic testing for melanoma, for basal cell carcinoma in particular, I think we're talking about really having an understanding of family history, and also the pace at which these cancers are developing. So, in the case of, let's say, Gorlin syndrome for basal cell uh, carcinomas, we're talking about people that are developing 30, 40, 50, hundreds of basal cells in their 30s and 40s. I mean, very unusual to see that in the general population for even for such a common cancer. Over 5 million diagnoses a year in the United States alone. So it's really a conversation between you, your physician, your nurse, about what makes sense for genetic testing, because sometimes it'll also involve other family members. It has implications for discussions with your children about risk, and so it's really ideally something that's taken um, uh, done in conjunction with genetic counseling as well, to help understand sort of what are the implications of a result, because it's not always clear cut, and you're looking for things. You may find things that are unanticipated. How do we digest that information, and what do you do with it, right, afterwards? So I think it's really understanding the whole picture of what you would do with that information, what can be done with that information afterwards. So I think it's very important to have that discussion with a team that you're comfortable with.
I'm getting questions fast and furious here, and kind
Okay.
of all along the same theme. And the theme seems to be, I've had a skin cancer, whether it's a basal cell or a melanoma. Should I get genetic testing? Or if there's been family members that have had it. And I think you've answered that in the sense that we talked about skin cancer is the most common cancer. If somebody has a spontaneous basal cell or squamous cell, often family members will have skin cancer as well because of similar behaviors and exposures. Your recommendation was people that have abnormal amounts, 20, 30, 40 basal cells or melanomas at a young age. That is not typical. So it's those types of situations where genetic testing would be recommended, because that's more likely to elicit a gene mutation rather than having a couple of skin cancers. We know people that have developed a skin cancer are at risk for developing other skin cancers. So that probably wouldn't prompt a reason for genetic testing, but certainly, you're nodding your head, so I presume that you're agreeing that
Yes.
it really is an individual discussion with your provider about whether or not it's appropriate. Any words of wisdom there? Different than what I said?
No, no, I completely agree. I mean, I think, it's largely, an issue of sort of the pace at which lesions develop. What have those diagnoses been? A little bit about, how your first degree relatives look, your children, your parents, in terms of, the pace at which they develop lesions, whether they have a lot of moles. But I think it's really a, a discussion that starts at that point, and not just, I had a couple of skin cancers and I need genetic testing, because, the reality is that the most important thing for most people is really being mindful about sun safety, knowing your skin, self checks, having people assist you in terms of keeping track of lesions that change in ways that, are concerning, and being able to get that lesion in front of a trusted provider, in your area, so that it can be evaluated formally.
Well, we are approaching the end of our time. I know I didn't answer all of the questions that came through, but I think our last two or 3 minutes of discussion did. And Pat had a question, and I would say to Pat, who has had multiple melanomas and dysplastic nevi, that would be a discussion with your individual provider based on risk. So I'll encourage you to talk with your provider about that. So I want to thank you, Dr. Tsai, thank the audience for participating in today's call. Lots of today's webinar, lots of great questions coming through, and this is certainly an area of interest. So don't forget to share this episode with any family or friends who may be interested in learning more about genetics and genomics. And please check out our website at ametSkinCancer.org for more information and resources. Lots available if you peruse the website. So again, to Dr. Tsai, thank you so much for joining me.
Thank you.
And until then, everyone, stay healthy.